It's a great pleasure. My name is Megan Campbell, and myself and Michelle will be moderating the webinar. And we have 3 panelists who've been gracious enough to accept our invitation. And as always, we have panelists who reflect the kind of breadth of our community. So we have a more senior researcher and as well as ECRs and mid-career researchers. So very lucky to have that range. I'll briefly just say a few lines about each of our panelists and then let them introduce themselves when they take the floor. And please do post questions in the Q&A as we go along, because we'll come back for a general panel discussion. So Dr. Dragan Rangelov is here with us. I apologize, Dragan, for butchering your last name. And he is a fellow in neuroeconomics at Queensland Brain Institute and also at the School of Economics at the University of Queensland. Dr. Sophie Lin is a postdoctoral fellow in the Cognitive Neuroscience and Computational Psychiatry Lab at Uni Melbourne, and also the facility manager for the magnetosec MEG there, and establishing the OPM-MEG facility too. And of course, Professor Tom Johnstone is also agreed to be a panelist, and he's the director of neuroimaging at Swinburne and also the Centre for Mental Health and Brain Sciences. We have links for people's quick access there in the chat if they want to look up a bit more about each of these panelists and their recent work. So yeah, we'll also try to post a poll at some point that will help us just get a feel of the audience, and then keep in mind that kind of breadth of audience that will guide our questions later as well. So yeah, I think that's fine. We hadn't really clarified who we'd invite to speak first, but we can go alphabetical and go with Dragan.
Sure, can you hear me?
Yes, we can hear you well, thank you.
Perfect. Being a nerd, I have prepared a short presentation, so let me just share my screen. And to provide some context, the topics I'm going to be talking about, given the fact that it's a short format, will not go into any degree of details about how exactly to analyze time series in terms of, say, EEG data or MEG data. The content of my talk is primarily geared toward maybe early PhD students or sort of halfway through their candidature, just to give them a flavor of what I find important in this whole area. So let me start by sharing my screen. Okay, so good. I hope you can see my slides. Yeah, so rather than going into specific details about nitty-gritty technical algorithms, how to analyze time series, I would just like to maybe share some thoughts about, say, values that I find important in guiding my research in using EEG/MEG. And I kind of, after some thinking, identified 2 such values. One being, let me just switch, one being reproducible research. I really try hard to make my research reproducible, and something that I decided to call economical research. Working at School of Economics, that sounded appropriate, but I will unpack this as well. So to start with reproducible research, the broad goal here is that anyone else on the world should be able to replicate my results. And when I was doing my PhD, which was some years ago, I remember using graphical user interface software, things such as SPSS and such, and I would never want to go back to those results because not even I could reproduce the figures that I have published successfully. Don't tell that to anyone, I guess. The bottom line is it is increasingly important that those results that we publish should be reproducible. And how can we achieve those goals? Well, there's a number of things that I have identified as important towards reproducible research, and I'm also going to present some tools that I'm using in the context of EEG analysis that are important for my research and achieving this goal. So obviously we would need to share our data. You have probably shared your data yourself as part of submission process, publication process. There's a number of such platforms such as OSF, Dryad, and a range of others. At UQ, we are fortunate to have local hosting platforms. Not every university is able to support that, but I personally find this to be very useful because ultimately the data that you collect belong to university, then to Australia more broadly, and only then to the world. The second thing that I also think is important is obviously to share your code. In order to share your code, you need to learn how to program. So I don't believe I need to preach that to this audience. Tools that I use in my work is, for example, GitHub, but there's a range of other platforms such as GitLab or Bitbucket owned by Atlassian, which is again Australian company. And also you can share your code using your local servers and custom-made solutions. In addition, in order for people to be able to reproduce your research, you would need to share the software that you used for analyzing data. Here, some things that are really important or popular are package managers. Conda is one particular flavor of package management, not the only one, but increasingly popular. You can also distribute the software in the format of virtual environments. So you can have lists of packages that you have used and kind of it's essentially a text file, and anybody can clone that virtual environment. And you can also engage in much more complicated and sophisticated methods such as sort of running whole kind of virtual computers or sort of kind of simulating the computing environment using Docker, using Singularity. And here I would also like to draw our attention to Neurodesk, which here at UQ we are quite proud of. It's a project under, I believe, management of Stefan Bollmann. But what I'm in particular proud of is that my colleague Angela Renton has contributed to Neurodesk a particular set of packages specifically tailored for analyzing EEG and MEG data. So you can have a look at that as well if you're interested in how can you make your research reproducible. Now, to talk a little bit more about actual data analysis. So obviously one of the important things in order to make your reproducible research is to use automated analysis pipelines. I again remember being a PhD student and analyzing EEG data, kind of spending hours in visual inspections and identifying every single artifact. And again, nobody in the world will be able to replicate those results because they would use their own individual kind of individually calibrated criteria to identify artifacts. So that's a problem. And in order to address that problem, people in fMRI world in particular have been pioneering such automated data analysis pipelines. I'm sure that you have heard of fMRI prep. In the area of EEG and MEG, there's also a range of different analysis pipeline. Particular flavor that I'm using in my work is so-called faster pipeline. You don't have to use that one. There's others, potentially better or worse. But one of the added values is that these automated analysis pipelines sort of are good for lazy scientists, in that you don't necessarily need to engage that much with programming specific processing steps or from continuous to segmented data. Of course, you would need to validate the outcomes because sometimes things can go wrong. So you come into that whole process towards the end of pre-processing stages, trying to see whether the pre-processed data makes sense or not. But the main idea is if you have automated analysis pipeline, you can just share that with people who want to reproduce your findings. Obviously, these pipelines will never be as good as an expert eye, but in that case, I think better collect more participants, increase your statistical power than rely on expert eyes, because expert eyes are few and far apart. So ultimately you want anybody from sort of a beginner to very sophisticated researcher to be able to reproduce your findings. And finally, I would like to mention BIDS, which is a mouthful standing for Brain Imaging Data Structures. This again originated in the brain imaging crowd, so I'm sure you have heard about it, but increasingly so BIDS are being used for all sorts of types of data. There's kind of working groups for eye data, there's working groups for EEG data, that's already quite well established. protocol. And one nice aspect of BIDS is that, well, actually two. One is that it kind of already tells you how to organize your files, because any starting PhD student always kind of faces that same problem, and each individual PhD student invents their own naming conventions. That's a waste of time, but in addition, that hampers sharing, because then the conventions for file naming and structuring are different from researcher to researcher. BIDS kind of standardizes that procedure, and you can look it up at this link. If you are keen to look more into that, a couple of years back, I have delivered a tutorial on BIDS as part of the OHBM brain hack. So you can check out my GitHub profile and you can see what we were doing there. Now, going further, so from this reproducible research going to economical research, at least my definition of that would be that one should analyze as much data that you have collected as you can. In other words, use it for the original purpose, but also reuse it shamelessly later on, potentially for entirely different exploratory purposes. And one particular aspect of types of such economical data analysis that is increasingly popular in literature is a set of multivariate analysis, which contrast very strongly to more traditional types of analysis, especially in the EEG literature, which I'm going to call the region of interest analysis. You probably, if you have ever read any paper on EEG, have heard of so-called ERP alphabet. So wherever you look in the EEG literature, there will be all sorts of acronyms, letters, numbers, telling you where in the on the scalp surface and in what time domain interesting things are happening. That's useful, but it kind of disregards that potentially interesting things could be happening simultaneously completely elsewhere in the brain. So just to illustrate the contrast between these things, so this is a set of my data from a particular study on decision making. We were recording EEG here. I'm plotting this sort of topographic distribution of activities across over 1 second period following a stimulus onset. And we see that things are happening all over the place. There's some positivities, there's some negativities. However, for this particular paradigm in the literature, people tended to focus on a particular cluster of electrodes that are marked here. I'm gonna plot the time course of those electrodes, and here we see that interesting things are happening. So I can go into details, but the bottom line is this kind of completely disregards what was happening in more posterior locations, more anterior locations. We just discard that information. Potentially more economical approach would be to use multivariate machine learning techniques to characterize the patterns of that activation across your experimental conditions. And we are using that in our work as well. So I'm showing here a particular set of results that used so-called population tuning strength analysis. There's different flavors to that. But the broad idea is that instead of analyzing just subset of electrodes, you're characterizing the whole pattern of activation. And as you can see, you can actually learn different things because the time course between these two types of analyses is quite different. And just my last slide, I would just like to mention a number of different types of multivariate analysis. So I already mentioned population tuning analysis. There's a whole host of different types of multivariate classification methods. And increasingly in the literature, people are using deep learning or neural networks modeling of the data. And each of these sort of flavors has kind of different software implementations. So you can find in kind of data analysis tools such as M&E Python or Fieldtrip code for maybe population tuning analysis. One of the popular toolboxes for such multivariate classification methods is a scikit-learn toolbox. And finally, for deep learning, you have things such as PyTorch or TensorFlow. Finally, if you want to actually engage in learning all these tools, I can recommend very warmly Neuromatch Academy. So if you haven't attended any of their kind of events, register whenever you can and whenever it comes. And they kind of keep very detailed archive of tutorials on YouTube, so you can also follow all the materials later on. With that, I would like to thank you for your attention, and I would be happy to continue discussion later on.
Thank you very much. Yeah, lots of great foundation points to start the discussion off with. So thank you very much, Dragan. Michelle, did you want to introduce the next speaker or pick who goes
sure.
next.
Next, introduce Dr. Sophie Lin. So Sophie is a postdoctoral fellow in the Cognitive Neuroscience and Computational Psychiatry Lab in the University of Melbourne, and also the faculty and also the facility manager for MEG there. So including the establishment of an OPM MEG facility. Sophie, could you please give up some talks and your perspective about the best practice of EEG and MEG?
Thank you for the introduction. So, I mean, in my part, so first, thanks for the organizer to organize this webinar, and it's a pleasure to present here. So I'll first share my screen. Is it presenting? Okay, so instead of talking about data analysis in my part, because this is one of a main focus of my work, like establishing OPN MEG here in Australia. So I'll be sharing mainly talking about these novel technologies and how it enables wearable MEG, as you might have already heard of it, OPN MEG. So I will first give you a brief introduction how this new type of sensor technologies detect tiny magnetic fields, and then the work that has been done to enable OPNs to be useful MEG purpose, and then talk about potential opportunities that this technology can bring to neuroscientific research. So we all know that the MEG and equipment so measure tiny neuromagnetic fields, but when we say tiny, so we are actually talking about millions to billions times smaller signals compared to the environment magnetic background fields. So that's the reason when you perform an MEG recording, so you have to put your scanner inside a shielded room. So that shielded room reduce the background field about 1,000 times smaller. So you're recording in an environment with about 10 to 100 nanotesla. So remember the number. And for the cryogenic MEG, we're using superconductor sitting in very cold liquid helium. So combining those two factors, you are having a state-of-art, the best spatial temporal resolution that a non-invasive neuroimaging tools can give you. But the use of liquid helium itself is actually also causing some issues. So to begin with, the sensors are so cold that you have to create a vacuum space that separates the sensors from the head. So that means the tiny signals you're measuring getting even smaller because the signals are inversely proportional to the square of the distance. And in the meantime, inside this scanner, so you have a fixed sensor array that's designed to be able to accommodate most of the head size, but are generally not optimal for individual heads. And that gets problematic for people with smaller heads, especially for children. And during scans, so people have to sit quite still, otherwise you lose the track of the special relationship between sensors and the brain, and you wouldn't be able to know where the signals comes from. So that's all the idea and also the problem we would like to make progress with cryogenic MEG. So imagine if we can get rid of the liquid helium and have a system that you can directly place on the surface of the head. That means you can get stronger signals. And you can design your sensor arrays to be flexible to best suit your research needs or different participant populations. And you can also allow participants to move a little bit more than they can do right now in a cryogenic system. So basically these things are now made possible because of OPN MEG. So the start of the OPN MEG, of course, they are OPNs, optically pumped magnetometers. But what exactly do OPN do? So basically, this is a standalone single channel sensors that you can see in this picture. Inside these sensors, you have an even smaller glass cells, which contains gas form alkaline atoms. They are magnetically sensitive. So when you place those atoms inside a zero magnetic field environment and has a laser line going through those atoms they will spin in a direction that's parallel to the laser line. Now, if there is a tiny magnetic field coming from the brain, which is not parallel to the laser line, it will cause the spin direction to change according to the directions of the magnetic fields. The important thing is those atoms, they have a tendency to get back to be parallel to the laser line again. And the way how they do it is by absorbing part of the laser line to realign themselves with the laser beams. And the magical part is that absorption curve is a linear function of the magnetic field, and that's how OPNs measures magnetic field. But the not so magical part is actually this linear relationship only sustained and at a quite lower range when it is smaller than 1 to 2 nanotesla. But remember, that is still 100 times bigger than the neuromagnetic fields that we are measuring, so which means it's sufficient. But in the meantime, also recall that inside a shielded room, you still have the 10 to 100 nanotesla. So that's basically too noisy for OPNs to be able to operate and to come up with a solution. So what the physicists do is they place some reference magnetic sensors inside the magnetic shield room, and using the magnetic field signals detected by those sensors, feeding them through very delicate design coils, calculated how much electric currents we need to place into those coils to counteract on those residual magnetic fields, and created quite nicely close to zero magnetic field, allowed OPNs to operate. So now you have an OPN MEG system. But what can you do with this OPN MEG system? To start with, we talk about you can get stronger signals. In fact, indeed, on the surface of the brain, you can get about 8 times and bigger signals compared to cryogenic system. So those enhancements basically decrease as you go deeper into the brain. But even at about 10 centimeters beneath the surface of the head, you can still get about 2 to 3 times bigger signals. So this advantage actually enables you try to detect some of the brain regions which are considered quite challenging for cryogenic system, like the hippocampus, or the cerebellum study that we did in 2019. And one of the reasons that the OPN MEG systems give you advantage to detecting those areas not only come from its proximities to the source, but also coming from that you can design your sensor array flexibly, best suited for the purpose of to detect your area of interest, like this temporal plus mouth sensor array for hippocampal detection, and also the inferior posterior sensor layout for the cerebellar detection. And to let you know that this flexible sensor layout is actually a very critical factor that for OPN MEG, it is particularly suitable for pediatric MEG study. And when it is needed, you can also place your sensor array in a quite dense way, allows you to perform more detailed cortical mapping, things like somatotopic or tonotomic mappings. And another advantage of OPN MEG is you now allow people to have a little bit more movement to be done during the recording. So which means you can perform more naturalistic paradise. One of the early classic example is basically the observations of beta desynchronization in the motor cortex, when people are playing little ball games during OP MEG recording in this 2018 Nature paper. And nowadays, there is also possibility you can do a little bit dance of hokey-pokey in the meantime, being able to see that OP and MEG record the corresponding motor cortex signals. But I have to let you know, OPNs are not without their caveats. So one of the things I do have to clarify is the current editions of OPN sensors at the moment still operate at a noise level that is higher than cryogenic sensors, even though most of the time, so these higher noise levels are quite well compensated by the proximity to sources, but there's something to remember. The other thing to remember is the current regimes of OPNs basically operates as magnetometers. So unlike the cryogenic system have both gradiometers and also magnetometers. So one simple thing to remember is that magnetometers, they are more susceptible to interference. So that interference can come from environmental noise that we talk about. In some cases, it can come from its surrounding sensors, and not unusually, actually, it can come from the experimental stimuli. So when you design an OPN MEG study, you have to be especially aware of whether you are introducing non-brain magnetic source by using your paradigm. And one additional thing I will have to remind you is that OPN themselves, at the current regime, they're not very good at dictating signals at very high or very low frequencies. So which means if that's the signals you're particularly interested in, you have to think twice before you decide whether you want to run an OPN MEG study or not. So in short, to run or not to run an OPN MEG study, so you have to choose a study that you understand you truly cannot do well with cryogenic MEG system. For example, like the paradigms are unlikely to be able to be done in a cryogenic MEG system, or participant who wouldn't do well inside a cryogenic MEG scanner. And on the ground, when you do your study, one of the things very important is actually to know your noise. So that's something you need to have a conversation with a medical physicist or engineers. Generally, they will be there inside within operating the study together with you. And that's something, the conversation you always need to have with them on the day of recording. So that's almost everything I want to tell you for today. Before I finish, I just want to let you know that we're currently building a full-head OPN MEG system inside this room in the campus of the Uni Melbourne, and it is expected to be operating in the second half of 2024. So hopefully in the near future, I will see you running your own OPN MEG study in Melbourne. So with that, thank you for your attention.
Thank you so much, Sophie. It's very exciting to see what's the cutting edge of things and that it's happening here in Australia. So yeah, well done setting that up and working on that. Yeah, so we have another panelist, of course, Professor Tom Johnstone, and would you like to take it away, and then we can have some shared discussions after that.
Yes, thank you very much.
Thanks.
And thank you, everybody. Thanks for the previous two talks too. That's terrific. And they actually said some of the stuff that I was going to say, so that's fine. It made it easy for me. I'll share my screen if I can. I'm on a old laptop because my new one is broken, of course. So bear with me here. Let's see, share. Trying to make this work. And I've got to start the PowerPoint presentation. Okay, and let's see if this works. I'm not seeing the presentation. Interesting. Just a minute.
No worries, take your time. I'll just encourage the audience as well to please do post questions in Q&A for any of the speakers as they're presenting, and we'll come together at the end.
All right, let's see if that works. Can people see my presentation there?
Looks great.
It looks.
Brilliant. Okay, good. And you're seeing the full presentation too, not the presenter here, right?
Yeah, just the slides. Yeah.
Okay, so that's a rather grandiose title, and actually in 15 minutes, there's no chance that I'm going to be talking about the future of all these things in Australia. And really the idea of this little short presentation, I think a little bit like the previous ones too, is to generate some ideas for discussion. This is something that I quickly whipped together today, and I thought was quite interesting. Now, this is not a really, really good analysis. This is a quick scan using Google Scholar of articles that either mention brain and fMRI, brain and EEG, brain and MEG. And you can see actually that although the articles are increasing enormously up until about the middle of the last decade, there seems to be a reduction in the number of articles published using these terms in more recent times. Now, that might have something to do with the way Google Scholar search works, but even so, I think it's very interesting to see that there seems to be at least a leveling off of the amount of research which is reporting fMRI and to some extent EEG. After that also increased over time. MEG, you can see, has never been as widely published as the other two, and that's got a lot to do with some of the things that Sophie just mentioned. I mean, it's pretty inaccessible, it's very, very expensive, and there aren't that many systems worldwide. That's had a slight uptick in recent times. And then, I don't know if anybody can see it, but right down the very, very bottom here, you can see a tiny new entry, which is OPM MEG, which Sophie just talked about. So maybe we'll see that column appear in the future. But I was sort of thinking, why might it be that there's been a leveling off in terms of the reported studies using fMRI and MRI and EEG? Now, of course, new techniques and new technologies always tend to have a wave of popularity, and then after a period of time, people start to realize that they have certain weaknesses. We've often seen this sort of diagram in many, many circumstances, pointing out the relative strengths of, say, fMRI in terms of time resolution and trade-offs with spatial resolution, with the EEG and MEG and so forth. And this is often used to justify the use of one technique or another. But I started to think about the way that these various techniques have been used. Dragan actually mentioned this a little bit in his talk too. And if we compare the way that fMRI and MEG have popularly been used in the past, fMRIs started out being used widely for block and event-related task activation studies. So trying to find out something about what was going on in the brain when people were doing tasks of various sorts. People moved on to studies of functional connectivity because they figured that, well, in tasks at least, perhaps it's the connectivity between different regions that matters. They also discovered resting state fairly early, and then that became important because it seemed to correspond somewhat to individual differences in certain types of traits and conditions. There were efforts to model what was going on in the brain during tasks with something like dynamic causal modeling to get at effective connectivity. And in more recent times, there's been a shift a little bit towards representational types of analysis using MVPA approaches, classifiers, representational similarity analysis, and also looking at a much more comprehensive look at connectivity using connectome modeling. Sort of in parallel, we've seen the use of MEG and EEG. I use MEEG to express them both. A lot of early research and continuing research using event-related potentials, looking at the alphabet as it was called, and the frequency domain alphabet. You're looking at the different frequencies and energy in different frequencies within the EEG signal. Event-related potentials quite often used to try to get at different aspects of task in a sub-event manner too. What components were making up a particular task performance or an event. Also efforts using DCM and similar techniques for effective connectivity, and more recently as well, representational analysis, and people starting to look at connectome modeling in EEG. I looked at this and I thought, well, one could summarize the two techniques, fMRI and MEG, in a rough way by saying that fMRI, its history has shown us that it tends to be good for neurobiology. Obviously, if we want to know where things are happening in the brain, it can tell us pretty well. And that might be very important for certain types of studies looking at, say, development of medicines, understanding neurobiology for different types of pathologies and so forth. But it's not so great for testing cognitive models because it really doesn't have the temporal resolution to tell us about the different components that happen in very, very rapid succession or sometimes in parallel when people are performing different tasks. And I think a large reason or a contributing factor to maybe the tapering off of fMRI, reported fMRI studies recently, is because people are realizing that it's not so good at testing cognitive models. And it's really hit its limits in terms of what can it tell us about the way certain types of tasks are accomplished. What are the computations, if you like, of the different steps? MEEG techniques, on the other hand, tend to be very good for testing cognitive models. They can tell us about the different subcomponents that go into performing a task or an action. But they're not so great for telling us about neurobiology. And that's more than just the spatial resolution trade-off, I'd say. It has to do with when things tend to be detectable with MEG. We have specific components that relate to specific events going on, computational events, if you like, but it's very hard to tie them down to neurobiology. So I guess all of that was just a way of highlighting the fact that when we're choosing a technique, we really need to be thinking, what's the overall purpose of the research we're doing? And I think we're starting to see now in the field a bifurcation, if you like, in terms of the way people are using these techniques and the sorts of questions people are asking. On the one hand, we have people looking at trying to understand basic cognitive functions, trying to test models of how basic cognitive, emotional functions, behavioral functions work. And then other ones which are more focused on neurobiology and using things like, for example, neural fingerprinting, trying to get at individual differences, trying to get at regularities perhaps in the connectome, in subgroups of populations, in psychopathology groups and samples, and so forth. The latter, which has really become the domain of fMRI and connectome modeling at the moment, requires huge amounts of data too. So we see a really concerted push to do multi-center studies, and we need to solve all the problems inherent in doing multi-center studies. Cognitive modeling, on the other hand, arguably, if you're trying to pull apart the very, very basic components of common cognitive tasks, you could argue there that you don't need such large numbers of subjects or participants. You can use a smaller N design, and that might be more appropriate, and then do repeated testing on the same subjects to get reliable measures. Okay, so I'm running out of time because I'm waffling too much. So I just thought I'd illustrate by an example of, well, I guess, sorry, I'm going to go back here quickly. I guess the point here too, though, is that what if we want to get at some components of both, right? We want to get cognitive modeling, testing some models of how some function is performed, but we also want to understand how neurobiologically that is supported in the brain. Well, that really, I think, calls for cross-modal analysis, the combining of these different approaches. And I'm going to give you one example here using EEG fMRI. This was a study that was published now a couple of years ago. Mareike Beyer from Germany, from Berlin, was the lead author on this one. And we were interested in testing models of processing of faces. And these are neurobiological models at some level, but they're also cognitive models. So they talk about the different steps in processing faces, visual information, how the brain computes what is and what isn't a face, how it recognizes different face features, different identities, and then more extensive processing of those face identities to do with who it is, our history with those people, how important those people are to us, and so forth. And so we use representational similarity analysis. I won't go into depth about this, only to say that here the idea is we're getting away from activation and we're looking at how information might be represented in the pattern of neural activity. And that pattern of neural activity might be as measured using fMRI or as measured using EEG. And here the idea is to combine both so that we can try to get an idea of when these stages, or test the model of when these stages are happening in processing the faces, and also where they might be happening in the brain. Another nice thing about RSA is that you can compare the measured representations from the brain, from fMRI or EEG, directly with theoretical representations, which we also did in this study. And the basic idea is this: you perform a task where you present the participants with different faces of different identities, perhaps expressing different emotions. And then you measure the brain activation using a standard fMRI bold sequence. And in each region of the brain, you look at the pattern of spatial activation, a spatial activation pattern around that particular region, and that's a vector. And then you have a vector like that for every condition in your experiment for all the regions around the brain. Similarly, with EEG, you measure the pattern of activity measured across your different electrodes, and at any given time point, you can have a vector of activity measured across different electrodes for the different conditions of your experiment. Now, what you can do then is make a correlation matrix effectively. We actually use a distance matrix, but it's conceptually the same thing between those two, between the different vectors of all the conditions in the experiment, to create this, what's called a representational dissimilarity matrix here, which shows how similar or dissimilar the representations, spatial representations are across the different conditions. And we do likewise for the EEG across the different conditions. Here for all the different parts of the brain, here for all the different time points post-stimulus onset. And then we can also construct theoretical models of how the activation should look if that particular region of the brain or that particular time point in the EEG is representing something such as the familiarity of the faces, whether the faces are of a loved one, or perhaps emotions that have been expressed by the faces. When you do that, you can regress your model dissimilarity matrices against the ones you measured from EEG, for example, across the time points. You can identify at which time points do the modeled RDMs, dissimilarity matrices, predict the RDMs that you see in the EEG, or explain them statistically. And as you can see in this particular experiment, there are a number of time points where there was significant prediction. And of course, that has to be corrected for multiple comparisons and so forth. And then, of course, you can compare directly between your modalities. So here we have the RDMs from the EEG at particular time points compared with the RDMs from the fMRI. And we can see, therefore, where in the brain the RDMs from the fMRI corresponded to the RDMs from the EEG at particular time points. And from this particular experiment, we were able to show that there was significant activation that was specific to aspects of the faces, not structural aspects of the faces, I should say, aspects of the faces related to identity as early as 108 milliseconds post-stimulus onset. And that was actually published around about the same time as this study by Nancy Canwisher and her group using MEG, but basically using a very similar RSA technique, similarity-based technique. The benefit of using this sort of cross-modal analysis is that we can combine representation, we can compare representations not only across modalities, but across individuals, because we can create representations over time or space in the brain for a child, for example, looking at stimuli, for an adult looking at stimuli. And here's where the MEG gains some of its advantages, because if you have an MEG child scanner, which they have in at Macquarie University in Sydney, it's much easier to measure MEG from a child using such a scanner than it is, for example, to do fMRI, or than it is to do EEG. But you can also combine representations from a whole lot of different modalities. So you can measure similarities or representations in terms of behaviours or subjective feeling states, in terms of physiological profiles, measured using psychophysiology, for example, or one thing we're looking at the moment is actually using MRI, cardiac MRI, lung MRI, and gastro MRI to get a representation of what's going on in the body in different emotional conditions. And at that point, then we can compare what's going on in the body with what's going on in the brain to understand perhaps body-brain connections in these sorts of these sorts of processes, I should say. I can see I'm over time, so I'll just finish up quickly by also commenting on something that Dragan said about the platforms. For EEG now, we do have a lot of extensions. As he mentioned, the University of Queensland group, especially Angela, has been very, very active in getting EEG processing software. Also from our group, Oren and David have been very active in contributing EEG and MEG processing software to this platform. Everybody can use it. You can either download and install it from the NeuroDesk website, or you can go to the ARDC's virtual desktop service. Every researcher in Australia has access to that now, and you can just fire up a desktop on their hardware, which is super easy. And I was going to talk about this, but I'm a bit out of time. This was just highlighting the fact that we now, through the NIFT, have plans to extend an EEG fMRI network across nodes across Australia, and potentially also in the future an OPM network, which would include and be spearheaded by Sophie's group at University of Melbourne. Thank you.
Thank you very much, Tom. We've got some hands clapping in the emojis down there. That was great. It was actually really helpful to see that map of where MEG and systems are now and where they might be coming in future, because yeah, the wider the access, the more research we can do together. So we have done the poll to ask our audience how much they currently use or have experience with these different modalities. And we've got 60% of people saying that they have used EEG, and nobody's saying that they only use MEG, but several saying that they use both. And there's a lot also who are novices to this area, including myself, although I couldn't vote. So yeah, 30% of people saying they've not used either EEG or MEG. So yeah, everything here is like a great opportunity for those people as well to understand more about the techniques and maybe how to reach out and collaborate on studies with those methods. I thought we might open the panel discussion actually by seeing if the panelists had comments or questions for each other, or things that they wanted to extend on having heard each other's presentations now.
Dragan?
Can I go? Yeah. Yeah, thank you to the other panelists. It was super interesting. I think one of the main, just to add to Tom's presentation and a little bit to mine, the main advantage of using such sort of multivariate data analysis techniques, and there's many, I couldn't even count them all, is that they kind of transform the nature of the measure from, say, an index of biological functioning, such as blood oxygenation or voltage on the scalp, into a different type of measure. So that lets you kind of compare similar things in a different space. So, for example, representational similarity analysis kind of lets you compare things in the space of the stimuli that you have presented. In a different type, say, in terms of population tuning analysis, it lets you compare things in how strongly is the brain representing particular types of stimuli, not necessarily as a index of the voltage or pulse signal, but in terms of feature specificity of that response. And that's really, I think, I can't stress how enthusiastic I am about that powerful way of transforming data, because it lets you kind of start to consider combining different methods, like in this lovely example between fMRI and EEG. Possibly it lets you compare formal models, like different models of similarity. And for me, this is the main kind of attraction or the selling point of such things. They're difficult to learn, but they're worth it.
I absolutely agree, Dragan. Thanks for that. And I think you're absolutely right. I mean, there are so many potential uses. You know, there's an enormous scope for the uses of those sorts of techniques to be expanded and applied to different problems. I got a couple of questions quickly. I just thought I'd quickly answer here that are related to that. What toolboxes do you use? That one, our study used the Cosmo MVPA toolbox. But there are a number now that exist in MATLAB and in Python, different toolbox for RSA analysis, for example. So, you know, it might be actually useful. I think there's probably a list of such toolboxes, but if not, I'd be happy to compile one and distribute or make a shared document. The EEG fMRI network is something that's in planning, and it's started to happen. We've got 4 nodes of the NIF at the moment equipped to the high density EEG and fMRI. One of the things I wanted to say about that, and also a little bit about what Sophie was doing, was that the idea of having such a network is not only to increase availability to researchers, but it's also to enable the sorts of larger scale multi-center trials and studies that are needed for certain types of questions, right? For looking at things like psychopathology or different types of population subsamples. And the challenge there for us as a community is going to be to create and build upon the work of people, for example, the COBIDUS OHBM group on MEEG, to build upon their work to create ways of making such multi-center studies practical and reproducible. So that has a lot to do also with what Sophie, I love what you said, Sophie, about understand the noise. That's going to be a really big part of this, understanding not only the noise within a site, but also the sources of variability across sites.
Sophie, did you want to take a chance to comment on the kind of collective things we've heard now?
I have to say, so I'm not, I wouldn't really say I'm an expert in terms of the data analysis, but one thing I do, I'm quite interested in and want to ask, maybe not fully related, is about, so a lot of the tools that we are seeing right now, maybe besides the machine learning tools are a lot of times still linear modeling. So what would you say in terms of, because the neuroimaging signals that we're measuring are basically quite complex signals. So what would you say about the pros and cons in terms of linear versus non-linear tools? So that's actually my question in terms of comment.
I can feel this question. Well, my take is linear models, we know most likely will not give you the real representation of what's happening, but they are useful approximation most frequently. So in that sense, I don't think that, I will be probably corrected, but I don't think that if you model something using linear model and find something, that result will be completely annihilated by using nonlinear models. So you can kind of approach the reality, however small, in however small steps, using linear models. Obviously, using nonlinear models potentially would be desirable, but I don't think we have common standards, common teaching methods. How do we develop pipelines? So I think there simply needs to be more work done in that space.
Yeah, you know, historically, linear models have done a pretty good job. You know, from a pragmatic point of view, they've done a pretty good job across domains that we think, well, they're probably not strictly linear. But the other thing that can be done sometimes is a transformation. So you can convert a nonlinear relationship to a linear one, and that's also a trick that's done quite a bit. And I don't know all the mathematical details of exactly how that's done, but it's quite a common trick in classifiers, machine learning, and so forth. There's something called the kernel trick, for example, which is used to do that sort of thing. So yeah, that is certainly possible. I'd be interested to know from other panelists, but maybe people in the community, what are the biggest barriers for people to actually do research using EEG, using MEG, or maybe combining those? Because obviously EEG is relatively inexpensive. So compared to the other modalities, it's something that a lot more people have physical access to, I would suggest. But I'd be interested to know there what the barriers are preventing them perhaps from using that more. And then obviously with MEG, there's barriers in terms of a lack of scanners, and hopefully that will be fixed in the future with the arrival of OPMs. Yeah, but maybe people can tell us something about that from their perspective. Or maybe the other panelists have some ideas too about what some of the barriers are.
I think I want to echo on Tom on that it's like that is definitely something like intense, because we are building this platform. We would very much like to know what kind of things that we can prepare in order to make the platform more attractive to the users, because the platform is only as valuable as from like the user's perspective.
If I may comment to this, I think the main barrier is the learning costs in general. So once you know those things and have physical access, I think everything is easy once you know how to do it. But the learning costs are, I think, substantial. And I don't think that we, as a community in general, have standards in teaching these things. Typically, starting to teach EEG or fMRI, people get exposed to that from PhD on, and they tend to be overwhelmed. And in addition to that, it's kind of left to the supervisors to organize the sort of curriculum and to monitor progress and you name it. I think Michelle here is in audience. I was fortunate enough to work with her and to follow her in that process, and as well as some other students. I think potentially working towards some attempt to maybe standardize the curriculum and what's important, what's not, what are good textbooks, which are not so good textbooks. I think that would be an important achievement in general, just in order to help people get a grip around all those very, very complicated things.
Yes, I agree on that point. And yeah, but actually there are many textbooks out there for EEG analysis, but now we see that all this advances in like techniques and all this analysis pipeline, we need to be aware of all other resources out there. So yeah, thank you all for sharing your perspective with us. And in the interest of time, I think that it's time for us to wrap up, and then we will have the AGM again later on. So
yeah, thank you so much. I was thinking, just while the AGM's getting going, I'll quickly make a Google document in that we can then share, so that you can all add in these resources that you're talking about, any useful websites, things that you've mentioned in your talk today that others might want to be able to open up again. So yeah, I'll just quickly make that and then share that. Okay.